The overall goal of the following experiment is to demonstrate sensitive and specific detection of high-risk HPV E6, E7 mRNA in tumor cells in head and neck cancers by using a novel RNA in situ hybridization technique called RNAscope. This is achieved by first pre-treating the tissue samples on the slides with heat and protease to permeabilize the cells to allow probe access. As a second step, incubate slides with an HPV probe cocktail targeting 7 high-risk HPV types, which hybridizes to E6, E7 mRNA. Next, amplify hybridization signals by hybridizing sequentially with the preamplifier, amplifier, and HRP conjugated label probe. The signal is then detected with a chromogen and visualized under standard bright field microscopy. Results are obtained that show HPV E6, E7 mRNA signals as punctate brown precipitates in HPV-positive tumors, but not in HPV-negative tumors.
The RNAscope assay uses a novel and proprietary probe design strategy for in situ hybridization. It allows single-molecule visualization and target quantification. This technology paves the way for researchers in the field of biomarker research and diagnostic assay development to identify and validate any RNA biomarkers discovered through microarrays or next-generation sequencing studies. Most scientists with previous experience